doing a day 4 warm up. And hopefully, like I said, you were supposed to do this yesterday. So hopefully this is just review, but I do want to go over one of these with you and remind you how to do a single trait or a monohybrid cross. So we're looking at, in dogs, there's a hereditary type of deafness caused by a recessive gene. Okay, so the recessive allele leads to deafness. So if we have 2 dogs who carry the gene for deafness, but they have normal hearing, so they carry the gene for deafness, but they have normal hearing, so they're heterozygous, what are the possible genotypes and phenotypes of their offspring and the percent chance for each? So we're going to talk today about percent and how we figure that out when we're looking at Punnett squares as well. Okay, so if these 2 dogs are normal hearing, they have normal hearing, but they carry the allele for deafness, that means they're heterozygous. So our cross is 2 dogs that are heterozygous for hearing. So filling in the Punnett square, just like we did last week, you're going to take 1 letter from the side and 1 letter from the top of a square, and you're going to bring them down together and write them both into the box. So these are the possible genotypes and phenotypes of the offspring of these parents. Okay, so we talked Friday about genotype ratio, which is counting up the number of puppies that would be homozygous dominant. That's always the first number in your genotype ratio. So the first number would be 1, right? Because if we look at the babies, there's 1 possible homozygous dominant. Then you're going to count up the number that are heterozygous for deafness. And there are 2, this square and this square. And then the last number is how many are homozygous recessive. And there is 1. On Friday, I told you, you don't have to write down the genotypes. You just need to write down the numbers. But what I've noticed in looking and grading homework number 3, people are making a lot of mistakes with genotype and phenotype ratio. So I think it is good for you as you're learning how to do this to actually write down the genotypes. All right, and then for phenotype ratio, there's only 2 phenotypes. You're either normal hearing or deaf as these dogs. So the first number you count up is how many are normal, which is the dominant trait. And there should be 3 here. There's 1, 2, 3. So these 3 puppies can hear because they have 1 dominant D, and that's all you need to be a normal hearing dog. So 3 of them would be able to hear to 1 deaf. Now, I want to quickly talk about something that students get confused. Students start thinking, oh, okay, so if these dogs had 4 puppies, 3 of them would definitely be able to hear and 1 of them will definitely be deaf. And that is not the case. Okay, what we're looking at here is a percent chance of the puppies having these genotypes and phenotypes. So if I said, what percent chance would a puppy be deaf? We have to look at how many squares out of 4 would have a deaf puppy. So if each of the 4 boxes all together equals 100%, that means each square is 25%, right? So if I said, what percent chance would this would these dogs have a deaf puppy, there's only 1 out of 4 chances. So 1 in 4 is what percent? Faith with conviction instead of a question mark. What is a quarter? 1/4 is what percent? Yes, 25%. I'm saying with conviction, this is not a question mark, right? This is 25%. Each square represents 25%. So if I asked you what percent chance would they have normal hearing puppies, what would you say? Good. I'm seeing a lot of 75%. Yep. So 75% of the time, these 2 dogs would have normal hearing puppies, but they have a 25% chance of having a deaf puppy. So if they could have 50 puppies and never have a deaf one. Every time they mate, they have a 25% chance of having a deaf puppy. That doesn't mean every 4th one will be deaf. Okay. When we do a Punnett square, we're looking at the percent chance of these offspring. All right. I'm not going to do the next 2, but you should do them yourself if you need the practice. And then if you have questions, need help, you can email us, you can come to morning office hours, you can ask at the end of the hour when we have office hours here. But you should feel comfortable doing mono hybrid Punnett squares by now. And if you aren't, there's a recap video from Friday, there's notes from Friday. But just make sure you don't fall too far behind, because now everything kind of builds on understanding this. Somebody's asking, we didn't have to write the percent, just the genotype and the phenotype ratio. That is correct. For homework number 3, I just asked you to do this, genotype and phenotype ratio. All right, yesterday you had a homework assignment that was assigned on Monday. And if you didn't do it, it's actually pretty fun. You're going to look at different traits and determine which one you have, whether you have the dominant or the recessive. And then you needed to pick 5 traits and take pictures of them, of yourself and your family members. So I'm not going to spend a lot of time on this. I was going to do this on Friday with you, but because of the snow day, everything got pushed. So if you need help on this assignment, I'm just going to say again, come to office hours or ask at the end of the hour today, and we can get you some help on how to do this. And at very minimum, we want you to do just your traits. This is a 10-point assignment, so you get 2 points for every trait that you do. Okay, there's 5 traits we asked you to pick. All right, so we are on day 5, Tuesday, February 2nd here. I'm going to skip this warm-up today because it's very similar to the warm-up we just did. It's looking at another Punnett square with a mono hybrid. And I want to get us to start thinking about dihybrids. So we're going to move on to genetics notes day 5. So first thing I want to point out here is there is a video linked here if you need extra help. I'm not going to show this video. It's about 6 minutes if you started at 6:51 and you watch it to the end. But if today you're like, I need to more explanation on that, this video is going to go over just the same thing that I'm about to go over. So you can watch that on your own if you need help. So today we're going to start looking at 2 traits at the same time instead of just 1. Okay, so we're going to combine 2 traits in the parents, and we're going to combine 2 traits in the gametes and 2 traits in the offspring. Okay. So I want to, I'm going to remind you that a parent only gives its offspring, its babies, 1 of each of its chromosome pairs. So it only gives 1 allele for a trait. So that stays the same. During meiosis, those 2 alleles separate. So if we're looking at 2 traits that Mendel would have looked at with his peas, because this is still what Mendel discovered, and how we know everything we know about genetics is because of Mendel looking at his peas. So we're going to look at 2 different traits. We're going to look at the shape of a pea, and I want you guys to use S. So there's a typo there. It says R. And we're going to use S because smooth is dominant over wrinkled peas. And it says round here. You need to change round to smooth. And we're also going to be looking at the color of a pea at the same time. And we're going to use the letter Y because yellow is dominant over green. And I know that might seem kind of weird to you because every pea that I've ever seen is green, but in Mendel's day, he noticed that yellow peas were dominant over green peas. So here's our key. So a dominant S is representing smooth. A little s is going to represent wrinkled. A big Y, dominant Y, is going to represent yellow, and a little y represents green. Okay, so these are the letters for our 2 traits. So we're going to start by crossing 2 parents that are heterozygous for both traits. So the cross is going to be big S, big Y, little y for both parents. Again, there's the R here, so you have to change that there's a typo there. Normally, I would not suggest using S's, but I found a really good example with S's, so that's why I changed it. But S is kind of a hard letter to use because it's hard to tell the uppercase from the lowercase. So we're going to do our best with that today. Okay, so remember that only 1 of an allele is given from each parent. So in a dihybrid, we need to include 1 version of the S. Sorry, this typo is still here. 1 version of the S and 1 of the Y allele in the gamete. So every gamete is going to needs to have 1 S and 1 Y. So how we determine the possible gametes that the parents can pass on, we use a similar method to math's FOIL method. So FOIL means first set, outer set, inner set, last set. So there's a little image of this right here. So I want to show you what this looks like. So if these are our parents, big S, big Y, little y. So they're heterozygous for both traits. 1 possible gamete that this parent can pass on is big S, big Y. Okay, that's the first possible combination. This parent can also pass on the big S with the little y. So that's the second possible gamete. This parent can also pass on little s, big Y. That's the third possible gamete. And this parent could also pass on little s, little y. So there are 4 possible gametes that this parent could pass on to their offspring. So I'm going to show you this one more time with the next parent. The next parent has the exact same genotype. So they can pass on big S, big Y. That's the first one. Big S, little y is the second one. Little s, big Y is the third. And little s, little y. It's because there are 4 possible gametes that we need to use a 4x4 Punnett square. Because remember before we were putting 1 letter for monohybrids because there was only 1 letter in each gamete. Now we have 2 letters in each gamete and we have 4 possible gametes. By the way, before we move on, I want to ask you, what possible gametes can this parent pass on that's homozygous dominant for both traits? So what's one possible gamete this parent could pass on?
SY, capital S, capital Y.
Yep, so capital S, capital Y is one. What else?
Capital S, capital Y.
Capital S, capital Y. Yep.
Do we just put that 4 times?
You would put that 4 times. Yep.
Okay, that's what I thought.
Yep. So this parent, because they're homozygous, they only could pass on 1 type of gamete. They're still going to make 4, but there's only 1 possible combination. Okay, so what about this parent? Big S, little s, big Y, big Y. What's one possible gamete?
Capital S, capital Y.
Yep, so that's 1. Yeah, Leah, so they would make 2 capital S, capital Ys, and then they would make 2 little s, big Ys, right? So the FOIL method you still use it for figuring out the 4 possible gametes. It's just if an individual is homozygous for one of the traits, they don't have as many different possible gametes. The heterozygous individuals have 4 different gametes. All right, so we're gonna set this up. And well, we're not really gonna set it up. I already set it up for you. So when we do a dihybrid cross, we use the 4x4 Punnett square because there are 4 possible gametes that the parents pass on. So remember that each gamete has to have only 1 of each allele, not 2 or not just a single letter. We have to have 1 of each. So here's how we would set this up. First, we would put 1 of the parents' gametes along the top. So we did that here. So we have our big S, big Y is 1 gamete, big S, little s, big Y, and then little s, little y. Okay, so that just came from above. And then we put the other parents' gametes onto the side. So here we have our second parent is along the side. And now we fill this in just like we did before. But this time we're going to keep our S's together. Okay, so always write your 2 S's first and then your 2 Y's second. And we still always put a dominant allele before a recessive allele, but this is of the same letter. Okay, so we still keep the S's together first and the Y's second. But we still keep a recessive behind a dominant for the same letter. So let's go ahead and start filling this in. So we do the same thing we did before where we take 1 gamete from the top and 1 from the side and we combine them to make that baby. Big S, big S, big Y, big Y. And then we move to the next one. Remember, we're keeping S's together. And then Y's go second. Dominant before recessive for the same letter. And then just keep doing that. It just takes a little bit longer, but the rules are the same as before. All right, so hopefully that part is not too challenging for you. Figuring out what the offspring would be. If you have all the gametes written, you should be able to fill in those squares. Just have to follow those new rules that you keep the same letters together. And I would always use the first letter of the gamete, so an S in this case, and then followed by the set of the second letter, Y in this case. All right, so if you scroll to the next page, we have this Punnett square actually filled in for you. So now we're going to take a look at the phenotypes. Remember, that means the appearance of the offspring. So we got to keep in mind that a big S represents smooth and a big Y represents yellow. So in this first box, I want us to find all of the individuals that have a big S and a big Y that represent being smooth and yellow. And I'm going to use an orange just because it's hard to see yellow when I write with it on here. But I'm going to count up and draw in all of my smooth yellow peas. So this first box is going to be smooth and yellow, right? Because it has a capital S and a capital Y in the phenotype. The next one is also has a big S and a big Y, so that's smooth and yellow. The third one also has a big S and a big Y, so smooth and yellow. And the fourth one also has a big S and a big Y. So the whole top row would all be smooth and yellow, just like the parents. The genotypes are not the same as the parents in all the cases, but the phenotypes are, and that's all we're looking at right now. So on the second row, the first baby is going to be smooth and yellow. The second baby is also, nope, second baby is not. It's smooth and green. I almost made a mistake there. So because there's two Y's here, this is not smooth and yellow. So moving on to the next one is smooth and yellow. The last one is not, it's smooth and green. Then I go to the next row. My first one is smooth and yellow. My second one is smooth and yellow, but the third and fourth ones are wrinkled and yellow. They have little s, little s. So on my last row, I have the first one is also smooth and yellow. And then as I look through the rest of them, there's no more smooth and yellows. So I'm just going to color these in yellow. And while I do that, I'm going to count them. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 smooth yellow offspring. All right, so the next one I'm going to count up is how many are smooth and green. But I don't want to accidentally look at these boxes again that I already filled in. So I'm going to use my amazing, super awesome scissors tool on my Notability, and I'm going to circle these P's that I just drew, and I'm going to copy them and paste them over on my second Punnett square. So I don't have to go through And I don't have to look at those boxes again because I already know they have round yellow or smooth yellow peas in them. So now I'm looking for the ones that are smooth and green. So this means they have a little y for green, but they have to have a big S for smooth. So starting here on my second row, I find my first smooth green one. And then there's another one at the end of this row, smooth and green. The next row that looks like wrinkled and yellow, wrinkled and yellow. Here's another smooth and green. And then we have wrinkled. So there are 1, 2, 3 smooth and green babies, offspring. All right, I'm going to use my handy dandy scissors tool again to circle these peas. I'm going to copy and I'm going to move it down to this Punnett square where I'm going to count up how many are wrinkled and yellow. And you might have to resize this one because this Punnett square is slightly smaller, but it works. So now we're looking for wrinkled and yellow. So I see a wrinkled yellow pea here because little s, and yellow, little s, and yellow. So I have 1, 2, 3 wrinkled yellow peas. And then if I use my scissors tool one more time, copy, paste, move my peas into place here. Then this last one is the pea that's going to be wrinkled and green. And this is the one that's recessive for both traits. Oops. Wrinkled and green. And there's only one possibility of having a wrinkled and green. One wrinkled green pea. So if we count up the number of peas that are dominant for both shape and color, we said there were 9. 9 were smooth and yellow. And then we count up the possible peas that are smooth and green. That was up in the upper right corner. We got 3. When we count up how many are wrinkled and yellow, we had 3. And then how many were wrinkled and green or recessive for both? There's 1. So when we put these into a phenotype ratio, we would write them as 9 to 3 to 3 to 1 because the ratio goes the dominant number first. So the number that's dominant for both. Then the number that are dominant for one trait, recessive for the next. The third number is how many are recessive for one trait and dominant for the other. So opposite what the second number was. And the last number is always the one that's recessive for both traits. So Mendel found this ratio to be true, 9 to 3 to 3 to 1, every time he crossed a pea, 2 peas that were heterozygous for 2 traits. And this helped him determine the ideas of independent assortment and that different traits are passed on independent of each other. And he wouldn't have been able to figure that out if he wasn't looking at two traits at the same time. So let's do one more example here together. So we're looking at two traits again, hair color and eye color. And it says here, suppose that black hair is dominant over blonde hair. So we're going to use big B for black, little b for blonde. And brown eyes, big E, are dominant over blue eyes, little e. So if we cross two people who are heterozygous for both hair and eye. So remember, we're looking at two traits here. So we're using B's and E's. So that means these parents are big B, little b, big E, little e. That's heterozygous for both traits. So big B, little b, big E, little e. So now to set up the Punnett square, we have to determine what are the possible gametes that each of the parents is going to pass on. So we have to do the FOIL method again. So if we look at our parents, they could pass on a big B, big E. That's a possible gamete. Big B, little e. And I'm just putting these up on the Punnett square as we go through because that's where they go, right? That's where we put the gametes. They can have little b, big E, and little b, little e. So those are our last two possible gametes. Because these two parents have the exact same genotype, they both could pass on the exact same gametes. So we just have to write these on the side as well. Once you have this set up, I don't think anyone has a problem filling it in. So take some time right now and go ahead and fill this in. Remembering you keep your B's together first and E's come second.
For the homework due today, can we
Yep.
just get a picture for ourselves, but like fill out all of them, but like not get a picture for all of them?
Do you mean taking just pictures of yourself and not any family members?
Yes, but filling like theirs in too, just don't have pictures above it.
Yeah, that'd be fine. At the very
Okay.
minimum, we're asking for you to take pictures of your own trait. So if you can't get pictures of your family, you know, you all have phones. You could ask for a picture of your mom's ear or your dad's ear or your sister in college. Tell her to show her tongue being rolled or not. But if you can't get pictures, don't stress, just do your own. All right, so once you have this all filled in, I just want to point out that we already know the phenotype ratio because it's going to be the exact same as before. So we could go through and we can count how many are dominant for both traits. And we would find that there are 9 of them. So we could go through and count them all. We just did this. But we would find 1, 2, 3, 4, 5, 6, 7, 8, 9 are dominant for both traits. Then we'd count up how many are dominant for B, but recessive for E. So we could go 1, 2, 3. And then count up how many are recessive for B and dominant for E. Oops, I should leave space for that. It would help if I made a recessive B. And we would find 3 of them. So right here, here, and here. And then our last number is how many are recessive for both. And that would be our 1. So it will always be a 9:3 to 3:1 ratio, phenotype ratio, when the parents are heterozygous for both traits. The next question is, what percent of the offspring will be totally heterozygous? So totally heterozygous means they're heterozygous for B and E. So you'd have to look through your offspring and see how many are heterozygous for both traits. And you'll find that there's 4 of them out of 16. So 4 out of 16 can be reduced down to 1/4. So it would be 25%. And then the last question is asking what percent of the offspring would be blonde-haired and blue-eyed? And that's this individual down in the corner here. So there's only 1 out of 16. So yes, there is math and science. So to find out the percent, you'd have to divide 1 divided by 16 times 100. You might need to get a calculator out to do this one. But a 1/16 comes to 6.25%. All right, so your homework for tomorrow is the next 2 pages. So you are being given a scenario of 2 parents. So you're going to have to first write down the genotypes of the parents and then their phenotypes. So remember, genotype means what alleles do they have? So we're asking what alleles does the female have? And they're saying she's totally recessive for hair color and eye color. Totally recessive means she's homozygous recessive for both. Phenotype is asking what does she look like? So this is where you give us words to describe what she looks like. So genotype is the letters, and she has 2 traits. We're looking at 2 traits here, hair color and eye color. Phenotype is what does she look like? And then you're going to do the same for the male who is heterozygous for hair color and eye color. So you're writing down what his alleles are and what he looks like. Once you have their genotype, now you got to figure out what possible gametes using the FOIL method they could pass on to their offspring so that you can set up the Punnett square. All right. I'm going to stop